तो कैसे हैं आप सभी? जिसको तूफ़ा से हो उलझने की आदत, जिसको तूफ़ा से हो उलझने की आदत, ऐसी कश्ती को समुंदर भी डुबा दिया करते हैं। तो इन्हीं लाइनों के साथ हम मूव करते हैं अपने डेली रूटीन वन लाइनर क्वेश्चंस ऑफ जेनेटिक्स। सो फर्स्ट क्वेश्चन इज दैट द color blindness the color blindness in human beings is a answer is sex linked character. Clear? Next question. Milking ability is cattle is an example of answer is sex linked characters. Clear? Next question. Gene present in Y chromosome and transmit from father to son are known as answer is haloendric. Next question. Bald head in human beings is a answer is sex influenced character. Next question. Next question is that human beings the type of sex determined is answer is XX and XY. Next question. The type of chromosomal aberration which suppress crossing over is answer is inversion. So before moving to next slide, we have to repeat all these question in very hurry. So first question is that the color blindness in human beings is a answer is sex linked character. Next question, milking ability is cattle is an example of answer is sex linked character. Next question, the gene present in Y chromosome and transmit from father to son are known as answer is holoendric. Next question, bald head in human beings is a answer is sex influenced character. Next question, in human beings the type of sex determined is answer is XX, XY. Next question, the type of chromosomal aberration which suppress crossing over is answer is inversion. Clear? Now we are moving to next slide. Next slide and first question. Next slide. So first question is that the chromosomal orientation during metaphase of meiosis is referred as answer is co-orientation. Next question. Plant with a genomic formula 2n-1 R. Answer is double monozonic. Clear? Next question. Inverted segment of chromosomes does not include centromere in. Answer is paracentric inversion. Clear? Next question. Next question. Expression of a recessive gene in hemizygous condition is known as. Answer is pseudodominance. Clear? Next question. Next question is that occurrence of more than two alleles at a given locus is referred as. Answer is multiple alleles. Clear? Next question. An example of duplication chromosomal aberration. Answer is par ice in Drosophila. Clear? So repeat all these question very quickly. So the first question is that the chromosomal orientation during metaphase of meiosis is referred as. Answer is co-orientation. Next question, plants with a genomic formula 2n-1 R. Answer is double monozomic. Clear? Next question, inverted segment of chromosome does not include centromere is in. Answer is paracentric version. Next question. Next question is that expression of a recessive gene in hemizygous condition is known as. Answer is pseudodominance. Clear? Next question. Occurrence of more than two allele at a given locus is referred as. Answer is multiple allele. Clear? Next question. An example for duplication chromosomal aberration. Answer is Barr eyes in Drosophila. Clear? Now we are moving to next slide. So first question is that the 3.3 the 3-point test cross the least frequency progeny is. Answer is double crossover. Next question. In the map distance between A and B, gene is 5, comma, B and C is 12, and A and C is 7. The gene order is. Answer is B, A, C. Clear? Now we are moving to next question. First question is that tendency of one crossover to reduce the chance of another crossover in its adjacent region is referred to as. Answer is positive interference. Clear? Next question. The map unit of linkage map are expressed in. Answer is Morgan unit. Next question. The chromatids of homologous chromosome pair are considered as. Answer is sister chromatid. Next question. Okay, so moving to next slide. Before moving to next slide, we should repeat again all these questions. So first question, in 3-point test cross, the least frequency progeny is. Answer is double crossover. Next question, if the map distance between A and B genes is 5, comma, B and C is 12, and A and C is 7, the gene order is. Answer is BAC. Next question, tendency of one crossover to reduce the chance of another crossover in its adjacent region is referred to as. Answer is positive interference. Clear? Next question. The map unit of linkage map are expressed in. Answer is Morgan unit. Clear? Next question. The chromatid of homologous chromosome pair are considered as. Answer is sister chromatid. Clear? So now we are moving to next slide, and first question is when three are no recombinant in the test cross progeny, the linkage is said to be. Answer is complete linkage. Clear? Next question. The chromosome number of human beings is 46. The number of linkage group formed in male are. Answer is 23. Next question. Question is, in balanced lethal system, the lethal gene are linked in. Answer is repulsion phase. Clear? Next question. Tendency of one crossover to enhance the chance of another crossover in its adjacent region is referred to as. Answer is negative interference. Next question. An example of a spontaneous mutation variety is. Answer is GEP-24 in rice. So repeat all these questions in very frequent. So first question, when three are no recombinant in the test cross progeny, the linkage is said to be. Answer is complete linkage. Next question, the chromosomal number of human beings is 46. The number of linkage groups formed in male are. Answer is 23. Clear? Next question, in balanced lethal system, the lethal gene are linked in. Answer is repulsion phase. Clear? Next question, tendency of one crossover to enhance the chance of another crossover in its adjacent region is referred to as. Answer is negative interference. Clear? Next question, an example of a spontaneous mutation variety is GEB-24 in rice. Clear? Now we are moving to next slide, and first question is that pseudo dominance is associated with. Answer is uh, deletions. Next question, unwinding of DNA double helix is induced by enzyme. Answer is Grace. Clear? Next question, directly strain weed like structure of the chromosome are termed as. Answer is chromosomers. Clear? Next question, a chemical that cause determination of DNA base is. Answer is nitrous acid. Clear? Next question, more than one triplet codon can code for the same amino acid, hence the genetic code is. Answer is degenerate. Clear? Next question, in an operon the site of transcription initiation is. Answer is promoter gene. Clear? So now repeat all these questions. First question is that pseudodominance is associated with. Answer is deletion. Next question, unwinding of DNA double helix is induced by enzyme. Answer is Grace. Next question, directly extend bead like structure of the chromosome are termed as. Answer is chromomere. Clear? Next question, a chemical that cause determination of DNA is. Answer is nitrous acid. Clear? Next question, more than one triplet codon can code for the same amino acid, hence the genetic code is. Answer is degenerated. Clear? Next question, in an apron the site of transcription initiation is. Answer is promoter gene. So clear all these questions. So now we are moving to next slide, and first question is that in next slide, splicing refer to the removal of. Answer is intron. Clear? Next question, DNA is a polymer of. Answer is nucleotides. Next question, the combination of nitrogenous base and sugar without the phosphate group is called. Answer is nucleotide. Riboside and riboside. Clear? Next question, the combination of nitrogenous base sugar and the phosphate group is called as. Answer is nucleotide, in कोष्टक ribotide and deoxyribotide, and nucleotide equal nucleoside plus phosphate. Clear? So now next question. Next question is the molecular formula of sugar present in chemical composition of DNA. Answer is C5H10O4. Clear? Okay, so before moving to next slide, repeat all these questions. Clear? Splicing refer to the removal of intron. Answer is intron. Next question, DNA is a polymer of. Answer is nucleotide. Clear? Next question, the combination of nitrogenous base and sugar without the phosphate group is called. Answer is nucleotide. Clear? Next question, nucleoside. Answer is nucleoside. Sorry. Okay. Now next question, the combination of nitrogenous base, sugar and the phosphate group is called as. Answer is nucleotide, ribotide and deoxyribotide nucleotide. Okay, you read that. Next question, the molecular formula of sugar present in chemical composition of DNA. Answer is C5H10O4. Clear? So now we are going to next slide, and next slide is first question is that the nucleotide in a nucleic acid differ only with respect to. Answer is nitrogenous base. Clear? Next question, the pyrimidine present in DNA are. Answer is thymine and cytosine. Clear? Next question is that the common purine present in nucleotide acid are? Answer is adenine and guanine. Clear? Next question. In case of RNA, pyrimidine base thymine is replaced by? Answer is uracil. Clear? Next question. In DNA, the ratio between the following base is close to unit. Answer is adenine and thymine. Clear? Next question is that Char Charg uh, e- Charles equivalence rule state that? Answer is A plus G equal to E plus C. Okay, so repeat all these question. First question is that the nucleotide in a nucleic acid differ only with respect to? Answer is nitrogenous base. Clear? Next question. The pyrimidine present in DNA are? Answer is thymine and cytosine. Clear? Next question. The common purines present in nucleic acid are? Answer is adenine and guanine. Clear? Next question. In case of RNA, pyrimidine base thymine is replaced by? Answer is uracil. Clear? Next question. In DNA, the ratio between the following base is close to unity. Answer is adenine and thymine. Next question. Char char graphs grass graph chargraph yeah pata nahi, I don't know what is the pronunciation of that, but I try to my best. Okay, so chargraph equivalence rule state that? Answer is A plus G equal to T plus C. Clear? So now we are moving to next question. Okay, so next question is that double helix structure of DNA was given by whom? Answer is Batson and Crick 1953. Okay, next question. The two standards of a DNA molecule are oriented. Answer is anti-parallel to each other. Clear? Next question is that each polydeoxyribonucleotide strand is composed of many deoxyribonucleotides joined together by phosphodiester linkage between their? Answer is sugar and phosphate reduced. Clear? Next question. The intron base are connected by? Answer is hydrogen bonds. Clear? Next question. The diameter of DNA helix in 20A, while it which the length of helix required to complete one turn is? Answer is 34A. Clear? So repeat all these questions. First question. Double helix standard was structure of DNA was given by? Answer is Watson and Crick 1953. Next question. The two standard of a DNA molecule are oriented. Answer is anti-parallel to each other. Clear? Next question. Each polydeoxyribonucleotide strand is composed of many deoxyribonucleotides joined together by phosphodiester linkage between their? Answer is sugar and phosphate residues. Clear? Next question. The nitrogen base are connected by? Answer is hydrogen bonds. Clear? Next question. The diameter of DNA helix is 20A, while its pitch the length of helix required? Answer is 34 angstrom. Clear? So next slide is okay. Sorry, I forget to add animation, but we read all these questions. So first question is that the hydrogen bond between the DNA strand break on heating of the DNA high temperature nearly 100 degrees Celsius. The process of separation of DNA is known as? Answer is denaturation. Clear? Next question. Reunion of the separate or denatured DNA strands on cooling is called denaturation or annealing. The optimum temperature for denaturation is answer is 20-25 Celsius. Clear? Next question. The conclusive proof that repulsion of DNA is by semi-conservative model was given by answer is Matthew Massillon and Franklin Stahl in 1958. Clear? So, next question. The replication of lagging strand generate a small polynucleotide uh, fragment called answer is Okazaki fragment. Clear? Okay, so now we are moving next slide. And first question is that single stranded RNA is the genetic material uh, in most plant virus such as answer is Mosaic virus. Yeah. Next question, double stranded RNA is also found to be the genetic material in some organisms such as answer is plant wounds and tumor viruses. Clear? Next question, prokaryotic mRNA molecule codes for more than one polypeptide. Such a mRNA is known as polycistronic mRNA. Clear? Next question, all eukaryotic mRNAs are coding for a single polypeptide specified by a single cistron. Uh, Such mRNA is known as answer is monocistronic mRNA. Clear? Next question, the amino acid is attached to the three end of the RNA that contains dash sequence. Answer is ACC. Clear? So repeat all these questions again in very frequently. So answer is single stranded RNA is the genetic material in most plant virus such as TMC tomato mosaic virus. Next question, double stranded RNA is also found to be the genetic material in some organisms such as answer is plant wound and tumor virus. Clear? Next question is that prokaryotic mRNA molecule codes for more than one polypeptide. Such a mRNA is known as answer is polycistronic mRNA. Clear? Next question, all eukaryotic mRNA are coding for a single polypeptide specified by a single cistron. Such mRNA is known as answer is monocistronic mRNA. Clear? Next question, the amino acid is attached to the three end of tRNA that contains answer is ACC. So clear? Now we are moving to next slide. एनर्जी, अपनी जो एनर्जी है ना वो ऑलवेज हाई रखनी है, कुछ भी हो जाए हमें पढ़ाई नहीं रोकनी, ठीक है? तो जैसा कि नेल्सन मंडेला ने कहा था, नेल्सन मंडेला जी ने कहा था कि एजुकेशन इज द मोस्ट पावरफुल वेपन you can weapon you can use to change the world. Clear? So this is my favorite quote. Okay, now we are moving to next slide, and next slide is and first question is that color leaf model of secondary structure of tRNA was given by answer is Robert Hole in 1965. Next question, in some viruses the DNA is synthesized from RNA in the presence of an enzyme reverse transcription. This process is known as answer is reverse transcription. Clear? Next question is that reverse transcription was first reported by answer is Tamin and Baltimore in 1917. Clear? Next question, the number and the sequence of bases in mRNA specifying an amino acid is known as answer is codon. Next question is that the set of bases in tRNA which base pair with a codon of mRNA is known as? Answer is anticodon. Okay, so repeat all this question. First question, color leaf model, color not color, okay. This is a clover leaf model of secondary structure of tRNA was given by? Answer is Robert Holley in 1965. Next question, in some viruses the DNA is synthesized from RNA in the presence of an enzyme, reverse transcription. This process is known as? Answer is reverse transcription. Clear? Next question, reverse transcription was first reported by? Answer is Tamin and Baltimore in 1917. Next question, the number and the sequence of bases in mRNA specifying an amino acid is known as? Answer is codon. Clear? Next question is that the set of bases in tRNA which base pair with a codon of mRNA is known as? Answer is anticodon. Okay, so next slide, and first question is that the set of all the codon that specify the 20 amino acid is termed as? Answer is the genetic code. And next question, a sequence of 3 nucleotide in mRNA code for an amino acid and hence the genetic code is? Answer is the triple codes. Next question, the 3 codon UAA, UAG and UGA do not code for any amino acid and hence they are called? Answer is nonsense codons, also known as stop codon or termination codons. Clear? Next question is that the codon AUG is known as? Answer is start codon or initiation codon. Clear? Next question, in most cases several codon code for the same amino acid, hence answer is the code is degenerated. Clear? Next question is one of the codons code for more than one amino acid, hence answer is the code is non-ambiguous. Next question, a subdivision of gene which act as a unit of function within a gene is termed as? Answer is cistron. Clear? Next question, a subdivision of gene which is the site of mutation is called as? Answer is muton. Next question, the smallest subunit of gene capable of undergoing recombination or subunit of gene which is the site of recombination is called as? Answer is recon. Next question, the enzyme responsible for joining of DNA fragment? Answer is ligase. Clear? So next question, in Drosophila phenomena the colonies of virulent strain are enclosed in? Answer is polysaccharide capsule. Next question, the experiment in which it was shown that DNA is the genetic material is bacteriophage is the? Answer is Hershey case experiment. Next question, the number of cytosine base in a DNA molecule is equal to? Answer is the number of guanine bases. Next question, quantitative characters show? Answer is continuous variation. Next question, a human blood group with antibodies against other group is? Answer is AB group. नेक्स्ट क्वेश्चन देखिए, हमारे यहाँ पे लाइट आ रही है, अभी अभी बिजली गुल हो गई है, लेकिन तो वो चिराग हैं जो अंधेरे में भी जलना जानते हैं, तो हमें अपना काम कंटिन्यू रखना है. तो नेक्स्ट क्वेश्चन इज दैट pseudodominance by a recessive allele is due to? Answer is deletion. Clear? Next question. Okay, so this is the table of ratio, F2 ratio, and genetic interaction ratio, gene action F2 ratio. ठीक है, तो duplicate dominant epistasis ratio is 15:1. Monohybrid F2 phenotypic ratio 3:1. Next, dihybrid F2 phenotypic ratio is 9:3:3:1. Next question, dominant and recessive epistasis is 13:3. Next question, monohybrid complete dominance is 1:2:1. Next question, dominant epistasis ratio is 12:3:1. Next question, duplication recessive epistasis is ratio is 9:7. Next question, recessive epistasis ratio is 9:3:4. This is table is very important. Okay, now we are moving to next. Thank you for watching. That's all for today.